is human cytomegalovirus, also known as HCMV. HCMV is a beta herpes virus with the genetic material surrounded by a capsid protein, which helps protect and enclose the genetic material. The capsid is then surrounded by a membrane that aids in the delivery of the virus to a cell. This membrane also contains spike proteins, which help deliver the virus to the cell. The genetic material is double-stranded DNA, exactly what's in our cells. One way that HCMV can transfer infection is through birth, from mother to child. The virus is a main cause of birth defects and can be dangerous for immunocompromised adults. If the mother has HCMV, 10% of cases will lead to the infant developing the infection within 6 months of birth. The infection can travel through the placenta, breast milk, or during delivery. HCMV is incredibly prevalent with a high seroprevalence. Seroprevalence is the percent of people that have immune responses to the virus, which indicates that they have had it in the past or that they actively have it. It is measured by using antibodies, which our body uses to fight infections, including viruses. The seroprevalence of HCMV is 60 to 90% globally, meaning over half the world has had this infection at some point in their lives. The virus can remain latent for years, leading to others becoming infected without presenting any symptoms in the original infected individual, which is part of the danger with this virus. One of the main treatments used for an HCMV infection is the antiviral letermovir. Letermovir works well for many patients with an HCMV infection. However, mutations in the HCMV genome have led to a growing resistance against letermovir, meaning the antiviral is no longer fighting the infection. This antiviral is really important for transplant recipients, as HCMV can be dangerous for these individuals, as most people are asymptomatic and therefore carriers. One specific mutation, where the glutamic acid is converted into a glycine at site 237 in the protein UL56, is associated with this newfound resistance. Glutamic acid, which is typically seen as glutamate, is a charged amino acid. Glycine is a small, non-polar amino acid, meaning it has completely different characteristics and can change how the protein functions. Protein UL56 is a terminase, which helps with packaging the HCMV genome. UL56 binds to the DNA, then binds to the capsid, where the DNA is stored in the virus. This protein, UL56, has been found to interact with another protein, UL89, in order to function in packaging the genome of the virus. So, what is our goal? The goal of this project is to express the UL56 mutated genome in insect cells via homologous recombination occurring in the baculovirus genome. When the virus is in the insect cell, the protein is able to express in that insect cell, meaning that everything that typically happens to the insect proteins will happen to the protein of interest, UL56, including modifications. Once we are able to express the protein, it can then be isolated from the insect cells for further analysis on the impact of the mutation and how it could infer antiviral resistance against letermovir. In this project, we worked through the process of getting the protein into the baculovirus genome so it can be expressed in insect genomes. The overall process is insertion of the plasmid into P-fastback dual, which will act as an intermediate for the transfer of the protein of interest genome into the baculovirus genome. Once it is in the baculovirus genome, the virus can be used to infect insect cells so that the protein is expressed in insect cells. Baculovirus is used because it's able to be maintained in insect cells, and the virus can infect additional insect cells and replicate with those cells. So now, for how we went through this process. The first step of our process was using PCR, polymerase chain reaction, which allows the amplification of the gene sequence of interest. PCR works by first heating the gene sequence so that the strands separate. This is called denaturation. Next, the temperature is lowered, allowing for the binding of the primer to the DNA sequence. The primer is then able to incorporate additional sequences, including restriction sites and tags, such as a his tag. The primer provides a start to the extension of the sequence. Then, that temperature is increased slightly for the extension of the primer, and the process is repeated to replicate the primers and obtain a large quantity of the desired DNA sequence. A restriction site is a site that allows for a recombination between sequences and allows the placement of a desired piece of DNA into the plasmid. Typically, the site is palindromic, meaning it's the same forwards and backwards. We use restriction sites to insert the gene of interest into P-fastback dual. The other piece added to the sequence by the primer in PCR is a his tag, which allows for purification of the protein following insertion into the insect cell. In order to confirm that the sequence is correct, it was run through a gel. The gel uses electrical current to separate the pieces of DNA from each other, and since we know the length of the sequence, we can estimate where it should be on the gel using a ladder with known molecular weights. Following PCR to replicate the mutated sequence of interest for protein UL56, the P-fastback dual factor, which is the in-between to get the sequence into the baculovirus genome to place into the insect cells, is ligated. This means that it is prepared for the insertion of the sequence. Following the insertion of the sequence into P-fastback dual, the cells are plated on LB/ampicillin plates, which are antibiotic plates, to select for the transformed cells, which should be the cells that have the protein sequence we are interested on it. Now that the insertion into P-fastback dual is successful, the sequence can be inserted into the bacterial artificial chromosome genome, again using those sequences that are repeated on both the P-fastback dual vector and in the back, similar to the restriction sites. Finally, the bacterial artificial chromosome will be inserted into the insect cell, where the protein of interest will be expressed and modified using the insect cell machinery, giving it similar post-translational modifications to what would have been seen in our own human cells, where the virus typically replicates. The goal of this project is to express the UL56 gene in insect cells, but what can we do with this after expression is successful? As I mentioned earlier, our protein of interest, UL56, is associated with letermovir resistance. This protein is also known to associate with another protein, UL89, in order to form a complex and package the viral DNA into the HCMV capsid. A future experiment could first isolate the mutated UL56 protein from the insect genome. Then we can also isolate UL89 after it's expressed in the insect cell and analyze if a complex is still made between UL56, the mutated protein, and UL89. Following this determination, we can add letermovir, the antiviral that the mutation confers resistance to, and observe if there is any reaction that takes place. By adding the UL89, we are able to analyze if the complex is still made or if the proteins remain separate. Then, the effect of the letermovir on the complex, or lack of a complex, can be analyzed. We would be able to determine if the antiviral is binding, not binding, and how it is functioning with the mutated complex. Based on the resistance seen in vivo with this mutation, we would expect that the letermovir would not bind. But we do not know the impact of the mutation on the protein's ability to create a complex. It is possible that letermovir only functions on the complex, or that the significant change in amino acid resulted in letermovir being unable to bind. Once we understand the mechanism of action of the resistance, we can potentially use this to create antivirals that can work around the mutated virus genome and maintain effectiveness against HCMV. HCMV is an incredibly prevalent virus in our society, and this research can give us insight into a major target for how to fight this virus and how the mutation is leading to resistance against letermovir, the leading antiviral to fight this virus.